And hello. This presentation should be pretty short. The chapter is short, the PowerPoint is short, so I'm going to go through that pretty quickly. Biochemistry. So the first section talks about organic chemistry being very important for life. We had this experiment by Uri Miller, and they set up this experiment to mimic early Earth's condition. They had electricity to represent the lightning that would have been taken place. And they put carbon monoxide, ammonia, water vapor, hydrogen gas. So all of these in these gases that were methane, right there, methane gas, that were in high concentrations within our atmosphere. We have a condenser, so you would have cold water come in, hot, the water would be warmer coming out, of course, because what it's doing is it's cooling down those gases into a liquid, and then it would accumulate down here. And then they could determine if those little cock stop right there, and they could then sample what was being cooked in this apparatus. Then we could also boil it and return it again back to the gas. And what they found was they honestly thought that it would take a long time for any kind of organic molecules to appear, if at all. And actually, it was a fairly short period of time where they started seeing some of these macromolecules that are required for life. So, organic compounds contain carbon atoms and are found in living things. Generally, we include carbon. We like to say like it's carbon and hydrogen, and that's what you see here. They have the two carbon atoms, and then these are all hydrogen. Okay, this is ethane. We say with hydrogen so that it excludes carbon dioxide or carbon dioxide. Those are not organic molecules. And if you look at their molecular structure, there's carbon dioxide, has no hydrogen in it. Okay. So most or inorganic compounds do not contain carbon. We really should say like carbon. I guess you could say just carbon atoms is your inorganic, and that's kind of the difference between inorganic and organic chemistry. Carbon bonding, we know carbons have four valence electrons, so they can form four bonds with other types of atoms. The carbon bond allows for carbon atoms to form a variety of simple and complex organic compounds. And we can see here, carbon can form a single bond molecule, a double bond. It can be what we call straight chain, or we have this branch. We can also form, and the picture here is kind of covering that. We can also form, like, so here's the straight chain, here's the branch. We could also get these rings, which we'll be seeing specifically with DNA and RNA, the sugar rings, which we did see in lecture already. We have what's called hydrophobic versus hydrophilic. And again, we've already talked about this a little bit. In a molecule in biochemistry is defined by its ability to dissolve or not to dissolve in water. So if it will interact or dissolve in water, we call it hydrophilic or water loving. If a molecule will not interact with water, it is referred to as hydrophobic or water fearing. So generally our rule is likes dissolve likes, nonpolar dissolves nonpolar, and we can go on and say polar will dissolve polar. And we can see it here. So what I have here is in the middle, we have these hydrocarbons, which are hydrophobic, and then you can see all the water molecules surrounding it. It's not dissolving. It's not dissolving in that water. Okay. That's why we have oil and water, they don't mix. So we have these things called functional groups. Functional groups usually influence a molecule's properties. It could have, you can take a nonpolar molecule and add these and we'll make it polar. So we'll talk about hydroxyl, an aldehyde, a ketone, carboxyl, amino, and phosphate. So the first one is a hydroxyl. You'll notice most of these are hydrophilic, water loving. It is an OH group. So when we have this dash here, we're saying that there's some, somebody could put an R for a side group if we wanted to. But when we look at this, like alcohols. As you know, alcohol dissolves in water, and that has to do with this little group right here, this OH. So you can see glucose with this OH here and how it's interacting with the water, which is why sugar dissolves in water. So it's nice having that hydroxyl there. The other one is a aldehyde, and that's where you have this carbon. R just means there could be carbon, carbon, more carbons on it. But on this molecule, we have pretty much at the end of the molecule, and I know this line here is deceiving, you have this carbon double bonded with the um, oxygen. And so that again is going to give us that hydrogen bonding. So it's going to um, make this molecule like this part, is really more nonpolar. But when we add this group here, it makes this end of the molecule polar. Ketones are very similar, except for you have this double bonded carbon in the middle of the molecule. And you can see here, right, there would be a carbon there, carbon there, carbon there, carbon there. So you can see how it's in the middle of this carbon and this carbon. So these molecules are showing you fructose. This is the structure of fructose. You guys all know you find that in fruit. Okay. Our carboxyl. Our carboxyl has this hydroxyl plus that double bonded oxygen. So we call it a carboxyl. It's polar again, hydrophilic. It can be amphipathic. We kind of talked about that. So when we look at this part of the molecule over here, that part is non-polar. We add this carboxyl group, and this end is polar, and this is going to be our fatty acid, which we talked about in lipids. Right. We can also see with those carboxyl groups how it can behave in water by losing, we can call that protonated. It gives off that proton, so it can go back and forth. This would be more acidic over here. This would tend to start going more basic. And then we have the amino acid structure. These used to be called peptide residues until we figured out the structure of it. And then we named it more according to the actual design of that molecule. So polar, meaning that it does dissolve in water. It is the main, this here is the main part of your amino acids. Right here, the side chain, what is hooked onto that central carbon is what determines which type of amino acid you have. And remember, there are 20 different amino acids. This group here is called the amino group. It's the nitrogen with two hydrogens. And then we have that carboxyl group here. And when we put that carboxyl in water, remember, it can give off the H plus, making the solution acidic, hence the term amino acid. We also have phosphates, and notice all the oxygens. Remember how I told you oxygens they'll share, but they don't share nicely, and so that makes them polar. They may also be charged because, again, sometimes we lose that hydrogen, so we can't get a charge on those phosphate groups. We will see this when we start talking about DNA and RNA. So here is that sugar group, and then attached to the sugar group is that phosphate group. We will also see this phosphate group when we talk about ATP, which is our molecule that gives us energy in our body. We already talked about, I want to make sure I didn't skip one. We already talked about this as well, but again, repetition is good. We talked about our condensation reaction. Remember, condensation meaning that we pull out a molecule of water. It can also be called, right, a dehydration because we're dehydrating that molecule. We're literally pulling this water molecule out in order to get this bond here. Remember, I called this bond. It is a covalent bond, but it does have kind of like a proper name. It's called a peptide bond. Okay. Now, when we break these polymers into monomers, ooh, I should clarify that. This is called a peptide bond if we're dealing with amino acids, right? Because we're connecting peptide residues. When you put in the water, right? We got to break that water apart, and we're breaking that water apart into oxygen, hydrogen, and then just the hydrogen, right? This molecule here would be your water molecule. We're splitting it apart so that we can break that bond right there. Oh, another word. Remember we talked about this being, since we're building anabolic. It's an anabolic reaction it's building versus this one being catabolic. So we're tearing it apart. Okay. So this is generally, we see a lot of this in digestion. Okay. And then the last slide is energy currency. So again, we have this molecule called ATP. ATP is made in the mitochondria, as you guys remember that the mitochondria, the mighty mitochondria, is the powerhouse of the cell. And that powerhouse is making ATP. Now, if you notice here, I have what's called ADP. ADP is called adenosine diphosphate, and it has two phosphate groups. We want to get to adenosine triphosphates, which has three, right? One, two, three. And in order to do that, we have to add energy. This is a type of reaction that, and we'll talk about exergonic versus endergonic, but those phosphate groups, they're charged. They're all negatively charged, and so they really don't want to be bound to each other. So we kind of have to force it, and the way we force it is by putting in energy. On the other hand, then when we take our ATP, we can break it, and when we break that bond, we are going to release that energy. And we're going to release that energy in order to do work. And so we're back to ADP. We go from ADP to ATP. So again, we'll spend a lot more time talking about this when we get to cellular respiration and photosynthesis. That is it. Like I said, this is a very short and sweet unit. Hopefully you've read it and you're working on the guided reading. And I will see you in the next video or in class. Again, as always, if you have questions, please feel free to ask.